Professor of Psychiatry here at UC San Diego. Uh, Michael, I might give just a very brief bio, if I may. And if I give a lot of fake news, could you please correct me? Uh, but Dr. Taffe completed his graduate work actually at UCSD and then set up a really vibrant lab over in the Department of Neuropharmacology at Scripps Research Institute. And of course, his work was focusing on the behavioral effects of drugs of abuse using preclinical models. Um, and then I was very fortunate as chair of the Department of Psychiatry to entice Dr. Taft to join our department. That was back when, in 2019, maybe, something like that. Anyway, and since then, he's established, again, a vibrant lab and very collaborative as well. His work involves many aspects of drug abuse using preclinical models, but of particular, I think, intersection with CMCR is his work on determining how THC and other cannabinoid compounds may alter liability for opioid seeking and consumption. And I believe that's going to be a theme of your presentation today. So thanks so much for joining us, and we really look forward to your presentation.
Thank you, Igor, and thanks to everybody else for showing up. Let me just share my slides here. Please speak up if you have a question in the middle of this. I'm pretty bad at monitoring monitoring your little pictures here, so feel free to say something. So I don't have any financial conflicts associated with the content of this, but I do want to mention that I'm going to talk an awful lot about vapor inhalation techniques, and we've worked closely with these on these projects with a vendor who makes this equipment, including a collaboration on a grant at one point, an SBIR grant. I want to acknowledge hopefully you can see my pointer, but a lot of this work was done by Jacques Wynn, who's now an assistant professor at Baylor University. So I'll be talking an awful lot about things he did. And he was actually the initial recipient of the award from CMCR as a pilot award, which I then had to take over once he was fortunate enough to get hired out of state. Fortunate enough to get hired, won't say out of state being the fortunate part. I'll show you a little bit of work from Arnold Gutierrez, who's now an assistant professor at UTEP. And then I wanted to point out that an awful lot of this work was done by Yannabel Grant on my technicians. Again, thanks to CMCR for the funding for some of what I'm going to be showing you. Our interest in this kind of started with epidemiological evidence, which is scanty, but suggestive, and it's the kind of thing where publications would come out in the context of legalization in either medical marijuana or recreational marijuana states, and looking at things like opioid-related overdose mortality going down, or at least a slower increase in fatalities, and also things like just general opioid use for pain. It's a little bit mixed, so there can be some additional studies which come out which seem to contradict original promising epidemiology, and there's always the caution about the sort of quality of epidemiological research when it comes to drawing firm conclusions. There has been some clinical evidence sort of parallel this. As you might expect, it's in the nature of pain usually, or analgesic use. And it's the same sort of thing you see with, in this case, trials, a reduction in opioid use, or the ability to even discontinue opioid use for a pain consideration, cessation or reduction, improved functioning, etc. And of course, being in real world, there are occasional studies which seem to indicate there may be some increases in harms related to opioids associated with cannabis use. And so, that's why one of the reasons we need controlled animal models where we can really focus on specific questions about how cannabinoids might interact with the effects of opioids. And, as Igor alluded to, the major theme of this presentation is going to be how do we interpret a change in opioid self-administration associated with THC? Just to sort of get our sort of start on this, methodologically, I'm going to be showing a lot of vapor inhalation stuff. So this is the e-cigarette devices which are being used to ingest cannabis as well as nicotine. And we just simply hook up commercial canisters to a sealed rat chamber controlled by a vacuum evacuation. And that's how we expose the animals to vapor. The first little bit of data I want to show you comes with a nociception or a pain assay. It's the tail withdrawal test, which you may be familiar with, where you stick the animal's tail into a beaker of 52C water in our case, and they tend to flick it out or remove it on the order of a couple of seconds. And so what you see here in this study is that we can sample them after vapor inhalation for a half an hour repeatedly. And if you give them the vehicle, which is PG, this tail flick response stays relatively consistent. If you give them a moderate dose of either THC or oxycodone by vapor inhalation, you see a slowdown in that response, an anti-nociception or an analgesic response. And if you add of course, it stays up for a longer period of time when it's THC, I should point that out. But if you add the two together in the same vapor exposure, you get this sort of additive effect. So this is consistent with the idea that, you could get reduction in opioid use associated with cannabis co-use if your endpoint was pain. Now, I do want to caution that when you sort of see these simple interactions, it may depend on the outcome measure that you're looking at. So in this case, what you're looking at body temperature responses. So THC, one of the canonical responses in a rat is a severe decrease in body temperature. And that's here. You can see in the females here, they have a substantial decrease in their body temperature. The males, it's not quite to the same extent in these conditions. But in our hands, heroin administration via vapor inhalation preserves a small elevation of body temperature across the session, a small elevation of the females and the males. And if you put them on board together, that's here. It's more obvious in the female group. The male one's hidden here. You get this kind of intermediate canceling out effect. And so it really does matter which outcome measure you're looking at as to whether you see sort of no effect, additivity, cancellation, whatever. This is not to sort of insult anyone's intelligence, and it's just a brief in the pharmacology. But the reason why you can have these variety of responses has to do with the pharmacology of the cannabinoid system. And so this is just a schematic showing you that the CB1 receptors are located presynaptically on terminals, and that they function, the endocannabinoid system functions as this kind of regulatory system on many synapses that use different neurotransmitters. And so, you have synthesis of the 2AG and the anandamide. It's this retrograde signal that goes back and modulates the signal of this incoming terminal, and whether they happen to be glutamate or inhibitory GABA terminals or other neurotransmitters, depending on where your outcome measure seems to be depending on and what systems it's depending on, it's no surprise that you can get this wide variety of interaction between cannabis and other drugs, in the case of opioids. Okay, so the real question here for us is, something like I just showed you, the lower use of two drugs to achieve the same clinical aim, does that reduce addiction liability? Or, even a non-clinical aim, say just getting high, does lower use of two drugs change that addiction liability? One very simple way to look at this is that pharmacological tolerance is dose dependent. And so potentially if you reduce the dose of two separate drugs, you may be producing less tolerance, which can drive addictive-like behaviors. But do keep in mind that the theory of, disruptions that are seriously related to significant addiction conditions tend to be final common pathway issues. And so it may not matter if the primary signals are coming in from different systems, if it's a final common pathway issue. I'll be telling you a lot about self-administration. Just to overview this, I know everybody knows this, but we have rats that are in an operant box. They have indwelling intravenous catheters where you can hook up to an infusion pump and make infusions of drug contingent upon them making a response on a lever. Simple stuff. This works for oxycodone. This is kind of a typical acquisition curve for a group of male rats, in this case, being trained to self-administer oxycodone intravenously. And you can see they sort of go up to this kind of asymptotic level, at which point they're relatively stable, and you can do all kinds of interesting studies. In this case, these animals were trained in 8-hour sessions. So this was our first study asking what can THC do about this intravenous self-administration of oxycodone in this case. What I'm showing you now are the intakes of oxycodone across a single 8-hour session if they've been exposed to that PG vehicle. And you can see across these 8 hours, their intake is relatively stable for 8 hours. If we've previously vapor exposed them to THC, we see a reduction in oxycodone self-administration as if we have this sparing effect. And it lasts about 5 hours after this initial vapor exposure. This is a replication, same group of animals, but just showing if you decrease the concentration of THC, you get a more limited response. And then showing there's nothing special about vapor inhalation. If you inject them with THC in more of a traditional model, you again get this reduction, especially for the first 5 hours or so of that self-administration behavior. Okay, so far so good. Looks like it's a sparing effect of having THC on board to decrease the desire to take oxycodone. Well, again, what does this mean? How do we really grapple with what the implications of this are? One of the things we do we did was this study here, where what you're looking at over here is under vehicle conditions or straight conditions. There's two separate per infusion doses of oxycodone, and this is kind of like a classic dose response. If you decrease how much you give them each infusion, they tend to take more infusions. Now, when we gave them THC, in these same injected doses I just showed you, basically, you get this stepwise reduction in self-administration behavior. But the interesting thing here is that it's parallel, depending on how much oxycodone they're getting per infusion. And this is inconsistent with ideas which our reviewer is very keen to advance, that THC just had this sort of generically disrupting or sedating effect on their behavior. And I would argue that proceeding in parallel like this, as if it's interacting with the dose of oxycodone you're giving them, is inconsistent with these more general proposals about what's actually going on. It's not a non-specific effect. Even more tellingly, to my mind, I just showed you fixed ratio, which is they just have to make one response on the lever to get an infusion. This is a paradigm progressive ratio where within the course of the session, each successive infusion of drug requires them to make more responses. And eventually the animals reach a break point where they say, no, I'm not responding anymore. And that's what we usually look at with a progressive ratio. And what you can see is that, yes, and I should actually point, if I didn't point this out, we'd switch into heroin at this point. So oxycodone train, this is for heroin. Increasing doses of THC reduces behavior, but importantly, at this lowest dose of THC, you got an increase in behavior. They responded more vigorously. They reached higher breakpoints for that same concentration of heroin in this case, if they had THC on board. And this is certainly incompatible with the behavioral suppression hypothesis. And traditional interpretations of progressive ratio is that what you've basically done is you've enhanced the reinforcing value of that unit dose of heroin in this case. And that's kind of our operating, conclusion at this point, is that, just a sort of spoiler alert, that's what THC is doing. It's enhancing the value of a unit dose of an opioid. Okay, so, that was the first thing. These are acute challenges. There are often a lot of other questions about, does this work chronically? THC does exhibit tolerance very rapidly. Can THC do anything to ameliorate an addiction-like state, whatever that might be, whatever you might be focused on? And, does co-administration the whole time that you've experienced an opioid change the chances of developing an addictive-like state? So we've done a little bit. I know you guys know this sort of theory, the negative affect theory of addiction. This is one of George Cube's famous depictions of the idea that there's a binge intoxication phase of taking drugs, a withdrawal or negative affect phase. But what's probably more important for understanding the rest of this talk is a figure he's included in other things, which show this kind of A and B process of elevation immediately consequent upon taking drugs, a discontinuation where the mood falls, and then some kind of recovery process where you go back to baseline. Of course, this allosteric hypothesis is that through these repeated cycles of intoxication and withdrawal, you get this change in the mood set point. And so the recovery to normal, so to speak, never gets back up to normal. The lows get lower and the highs don't get as high, maybe just crest regular mood, or in some cases, don't bring you back to normative mood. One colloquial way of thinking about this is they're just taking drug to get straight. Okay, so how does that interact with what we've been doing with THC? Well, before I get to the THC part, I'm going to talk to you a little bit about, a little side view on this kind of dysphoria measurement with oxycodone self-administration. This is Jacques Min's work. So how you can, one of the ways you can assess sort of this hedonic state, if you use affective state in a rat, is with the intracranial self-stimulation reward paradigm that's been worked out in the context of these models by Athena Marcoux back in the day and her groups. And the idea is that if you put a small electrode into areas of the brain and deliver current, it has a reinforcing effect in a rodent. You can make them work for current. In our case, we use a thresholding procedure, which is that you can ramp that current up and down across the session, and the animal can be trained to tell you when they feel it. And so they give you an idea of what their threshold is for feeling the current threshold for feeling a reinforcing effect. And the idea with this is that if those baselines are stable, which they are, if you do anything which causes the reward thresholds to go up, so they require more current to say they felt it, you interpret this as an anhedonic state. And if you have a decreased current threshold, you interpret that as being a hedonic state. These are some of these older studies from Coombe and Marcoux and Paul Kenny and Serge Ahmed. But as I was saying, those thresholds, that's the B panel. In case my pointer's not working, the B panel shows the incredible stability of these thresholds over time in untreated animals. The C panel shows you that if you measure before and after a short access session, a 1 hour session of heroin self-administration, they're a little bit elevated before you give them drug. If you give them heroin, they're a little bit decreased. It's about what you see. But in these sort of escalation models where animals have 23-hour access to heroin self-administration, not only does their, in the A panel here, not only do their number of infusions go up instead of staying stable, but those reward thresholds increase too. And so this was always taken as one of the pieces of key evidence that what we're seeing, this driving of the behavior, was an increasing dysphoric state. That was back in the day. All right, so this was Jacques' work showing that with extended access to oxycodone self-administration, you do get this kind of escalating pattern of behavior in the long access ones. These are 12-hour sessions versus a 1 hour session short access animal. So it works for oxycodone. You may notice the thing we notice, which is since I don't like to beat my lab and I'm lazy and we run weekdays and not weekends, he noticed this jump up pattern. And this was kind of interesting. And the reason this bump up pattern is sort of interesting, because you think about the theory of the negative affect theory, it's that escalating drug intake is driven by withdrawal, basically, negative affect. And we know that, in various withdrawal models that withdrawal builds in rats and other organisms, it builds over the course of days and not hours after the last drug exposure. And so by stretching this out to 60 hours as opposed to the usual 12 hours, what we were doing basically was, we infer, is testing this idea that an increasing withdrawal state should lead to increasing intake. So that was what we noticed first in the behavior, and we spent some time trying to parse this effect. This is a replicate group of animals. You see the same kind of basic weekend-related pattern. We actually started doing things like omitting a day in the middle of the week to make sure there wasn't anything really funny going on in the vivarium during the weekends that we weren't thinking of that was doing this. No, it was this discontinuation. And then it ends up being relatively stable in terms of their intake after a while, and they stopped doing that. Now, these animals were implanted with ICSS electrodes and trained. And so we saw in those animals that across that first week, they were going up in their thresholds in a regular stepwise pattern, precisely like we would have predicted. But unlike what we would have predicted, they reset across the weekends. Now, I'm here to tell you, we still don't know exactly what this is and why this is inconsistent with the way we're thinking about this. But the point I want to make is that this is a very consistent pattern across weeks of steady self-administration behavior in 11-hour sessions, not 12-hour sessions. We need an hour to test them at ICSS, but it goes up, it goes up. There's that week where we intimated a single day, and so you see it starting to come back down again, and they restore, and then this is an abstinence week. And so it's very titratable, I guess I should say. And this is another group, another replicate sample, just some treatments in here that I'm not going to talk about, but suffice to say, this is like sort of out at day 20, and what we're doing here is just showing that it was more or less similar to this, where we had them up for these sort of 5 days up, the reset, and then we just sort of extended it for another 5 days to make absolutely sure they would stay up. And then we introduced the saline self-administration. We saw them step down, continuing this pattern. So we actually watched it drop down. And then again, the idea this is titratable. If we give them 1 hour oxycodone sessions, there's this kind of very small response, particularly in the control group. Again, I'm not talking about what the experiment is here. If we substitute heroin and go back to 11-hour sessions, you restore the same pattern that was seen with oxycodone. And if you drop that to 4 hour sessions, you have this kind of intermediate response. So it's very sensitive to what the self-administration conditions are that you're exposing the rats to. So that's where we get to test THC. And so what we did is in one of these groups, actually the last one I just showed you, we asked what happened if we give THC before their self-administration sessions. And so here in the A panel, what you can see is this is their infusions across 4 successive days. It's a crossover design, so all the animals are getting either vehicle or THC. And THC reduces the amount of oxycodone they're taking in these 6 hour sessions, just the same way as it did in the first study I showed you. And of course, the reason we cut this down to 6 hour sessions is because I actually showed you that the effective THC really only lasted about 5 or 6 hours. And so we thought it'd be a little bit troublesome to try to figure out how to do that across 11 hours. And the key for this is the dysphoria is exactly the same. So even though they're taking less oxycodone, this titratable response is doing the same thing, as if whatever the disruption associated with the intoxication and potentially the withdrawal had to do when they had THC on board and were taking less, ended up having the same effect on this measure of affect. Okay, now we're cooking with gas. That brings me to the work that Jacques proposed for the CMCR award and that we conducted and finished under that. And so those were, again, that was sort of semi-chronic, 4 days of THC in trained animals. We wanted to know what happens if you give the THC right from the start, if they've only ever experienced oxycodone in the context of THC versus a vehicle injection. What you can see is in this group of animals, so this includes both males and females, where half of the animals were given a THC injection before the sessions and half the animals were given a vehicle. And I hope you can see that they take less to about the same amount as that 4 day study and about the same amount less as I showed you in the single acute things. And it persists throughout acquisition. Now, the story gets a little more complicated than that. So this is the combination of the males and the females. The males and the females were a little bit different in the sense that the males started off lower, they have THC on board, but they sort of collapsed back towards their vehicle-treated cohorts until we gave them a higher dose. So we're sort of interpreting as a tolerance effect. The females stayed down for longer and maybe, increasing the dose kind of persisted that a little bit, but no big deal. This is a little bit unusual in the context of rats. One thing you may not realize or may not know is that for most outcome measures related to sort of non-contingent THC in males versus female rats, the females tend to get tolerant to a larger degree for the same dose, or they get tolerant with a less dose or lower exposure. They seem to be more sensitive on a number of outcome measures. This is a case where if this is tolerance, it looks like the males are sort of becoming more tolerant to whatever this is. And so again, it's a reminder that things really depend on outcome assay. Okay, so why are these groups 2 and 3? Because we started with a group of males who were implanted with ICSS transmitters, and we saw as in that first week, we saw this difference associated with the THC pretreatment. We saw them collapse back together, which is why we increased the dose here, because we were already thinking about tolerance. We ran this group. But here's the interesting thing, this little bump up here. Now, these are 6-hour sessions, and our sort of weekend jump up effect tends to depend on how long they have self-administration. We see it most robustly in 12-hour sessions, and we see it less often and less consistently, say, in an 8-hour session group of animals. But, mother's eye, you can kind of see it there. Well, this was interesting because they're actually taking less drug, and they have this larger weekend bump up effect. That's why we ran straight through for these other 2 cohorts. You didn't see anything like that. There's nothing magic about this first weekend. And until we gave them a weekend, and we give them a weekend, we saw this recapitulated in these groups as well. And so again, if we think about what we are thinking about and leaving the ICSS a little bit aside, that this is somehow related to, let's say, the severity of the drug taking, the severity of the intoxication phase, maybe. It's interesting that it appeared in the THC group. Because again, our working hypothesis at this point is that what we're doing with THC is the reason why they're taking less oxycodone is because you're increasing the reward value of each one of those infusions. And if so, you can see how this might be an interpretation that they're coming back up to the same intoxication level, if you will, as the animals that are not on THC. Okay, so this is that group one. This is their the same infusion data now broken down by week, just so I can highlight a couple of things. One, this is that first week where they separated from each other. They kind of closed together for the second and third weeks. And then in the fourth week, what we did is we switched them over. And so the animals that had been on vehicle were switched over to THC, and the animals that had been on uh, THC were switched to vehicle. And so we sort of recapitulate this separation when we did that switch. These are the ICSS implanted animals, and you can't see the pattern quite so well, but suffice it to say, we have this sort of increase across the week, a little bit of a recovery on the Monday, and a reinstantiation. And the important thing is that during the switcheroo, there's no effect here. Even in that initial week when they're taking significantly different amounts, if anything, the THC animals were the ones that their thresholds were going up. It's not statistically significant. They're not really different, but they weren't less, let's put it that way. And again, consistent with this idea that even though they have less drug on board, they're self-administering less oxycodone, whatever the sort of, reinforcing and therefore the dysregulating, the negative affect that builds up across that week is the same, if not greater. So these groups of animals, now this is a ways out. We had the initial acquisition, we had the switch, then we had some discontinuation and some dose response. And so basically both groups of animals had experienced THC before their self-administration and vehicle before their self-administration. And if you look at the collapse across all the animals, what you see is that if we inject them, this is a progressive ratio test. If we inject them with THC before the session, they take more, they reach higher breakpoints, I should put it that way, than under vehicle or no injection conditions. And this pattern is recapitulated in both of these subgroups, the ones that started on vehicle and went to THC, and the ones that started on THC and went to vehicle. So it's probably, fairly consistent at this point across these animals. So this is a little bit new in the sense that this was oxycodone instead of heroin, which is the thing I showed you in the first case. And these are against a background where they've had considerable amount of THC plus oxycodone exposure. So you see the same effect, basically. They're willing to work harder for a unit dose of oxycodone. Okay, what about CBD? We have done a little bit of work with CBD. And so this is the group 3, I think it was, the female animals. And it was the same kind of approach where they had been trained under THC versus vehicle. They'd been switched over. They had an extended period of experience. And so I'm not going to be showing you these 2 different groups for this. But again, they're self-administering in 6-hour sessions. This has now been brought down to 3-hour sessions because that's what we use for PR. And so this is vapor inhalation of CBD. And this in the first A panel here, what you see is their number of infusions they get of oxycodone are reduced by CBD inhalation prior to the session. That's under fixed ratio. We didn't see a significant effect of THC inhalation in this group, maybe due to sort of their chronic THC experience and tolerance. We actually went and injected them so we could, get higher exposure levels. And sure enough, we give them enough THC, it does reduce their responding for oxycodone under fixed ratio conditions. So what about the PR? Because we sort of talked about the PR as being special. This is these same animals, these female rats, being exposed to either CBD vapor or THC vapor. This is their breakpoints under a PR procedure. And 2 different concentrations of oxycodone as a reinforcer. Now, we didn't see much difference between those for reasons I'm not really understanding, but suffice it to say, again, this is now THC vapor in these female animals, increases how hard they're willing to work for oxycodone versus vehicle or even versus CBD. And we didn't see any statistically significant change in their responding for oxycodone after CBD exposure in this progressive ratio. So it's only under those easy access conditions that it decreases things. It doesn't do much under progressive ratio for these animals. And we were a little bit concerned, we do these longitudinal studies session after session. We're always a little concerned about plasticity, tolerance, if you will. We don't know much about what vaping CBD does to these animals in a repeated situation. And so we went back and did the same examination under FR. And now we have 2 different concentrations of CBD, but this is the one we used originally. And sure enough, we were able to see a reduction in self-administration behavior under FR conditions with CBD vapor. So we think they're probably doing something different, not too surprising. Um, so sort of I'm almost, yeah. Uh, uh, the it's pretty clear that THC can reduce oxycodone self-administration, whether it's acute, whether it's chronically, every single time. It increases the breakpoints for opioid self-administration or progressive ratio, which I think is, I think is super important here because we canonically interpret these changes in responding on a progressive ratio as if that is what they're willing to do, as if it's a reflection of the value of that reinforcer to the animal. If they're going up under THC, we have to interpret that as being an increase in the reinforcing value of unit dose. Um, I kind of wrote this THC does not ameliorate dysphoria a little bit wrong, but basically that had to do with the ICSS in the sense that if you have the THC on board in self-administration of oxycodone or heroin, it doesn't seem to do much to the dysphoria that's generated, particularly under long access conditions. And so, again, this sort of gets at this idea that putting them to two basically lower amounts of two drugs together reaches the same endpoint as just the opioid itself. Uh, that intermittent discontinuation, it's a longitudinal thing, and we only really see that in acquisition, so it's a little bit hard to do a lot in terms of testing that. But it certainly seems to be the case that, the first week or two of acquisition of self-administration, whatever that little bump up is with 60-hour discontinuations is enhanced by THC, despite the fact they're taking less oxycodone leading into that. And then CBD, in contrast, might reduce self-administration in a manner that's, inconsistent with what THC is doing and maybe more consistent with sort of a therapeutic effect and more of a generic blocking effect. I have time to talk about a little something else, which is somewhat related. Uh, and this is again one of Jacques' studies. Um, so what I've shown you is what happens in the presence of, basically CB1 signaling in the presence of an exogenous cannabinoid. What happens? Well, I'm going to describe to you now is what happens when you have diminished the effect of the endocannabinoid system, in this case by repeated exposure or developing tolerance. Um, this first slide just to show you, Jacques did this with Adam Fields in the department, just to show you if, in our paradigm, if they have twice daily THC vapor exposure sessions for 10 days, it reduces CB1 expression. So we do have, some backup for the theory, but of course, being the behavioral pharmacologist, I'm much more interested in the fact that we see this in terms of pharmacological tolerance as well. So this particular set of studies, adolescent rats in this case were exposed twice daily to vapor for 10 days, vapor of THC or vehicle. And so I showed you some indication of this before, but basically THC, particularly vapor exposure, decreases the body temperature of rats. It's a very systematic and consistent effect. So I'm showing you here is two groups of male and female adolescents exposed to THC vapor. It drops their body temperature, reaches a nadir at 60 minutes. We didn't see the 30-minute point in these animals, which is why it looks a little different, but 60-minute nadir is really the key here. These animals eventually, so after that single first challenge, were devoted to either PG repeated exposure or THC repeated exposure. These are the THC repeated exposures during adolescence. You see that body temperature response. Here's that first one in the THC groups, males and females. It stays down for a few days under repeated exposure. You get a little bit of tolerance in the females. This is somewhat what I was referring to about females becoming more tolerant more rapidly. You go into week 2, the females are still a little bit tolerant and the males are down. This is the end of adolescence, 94, at 46 days of age. This tolerance persists. And so this is now young adulthood, day 85 onward. And what you see is this is now the challenge in both groups of animals. The green ones are the ones that had this repeated THC exposure and developed this tolerance. The blue ones are their PG exposed companions. And what you see for these female animals is there's that tolerance at intermediate concentrations. And if you raise the concentration high enough, you can see the whole response, classic tolerance. Interestingly, the same pattern was seen in the males, which intermediate doses, you see tolerance in the repeated exposed THC animals, and you saw, the full effect in their PG exposed compatriots. And this is, again, this is like 40 days after they've last seen any drug. Okay, so that's the kind of setup for this paradigm doing this to the animals, and that more or less lasts forever. We did this and asked, what about self-administration? And so adolescent exposure twice a day for 10 days. We come back later in adulthood and train them on oxycodone self-administration. These are the females. This is their infusions. This is their lever discrimination. Not much difference between the groups. Not much difference associated with that adolescent exposure. We probe them with an FR dose response, a PR dose response. Not much difference between the two treatment groups. And the same thing was seen in the males. And so the males, again, self-administration right on top of each other. They learn at about the same rate, and their dose effect curves look about the same. But here's the thing. In the females, at the end of the days, and this is at PND 200, so long ways out from their repeated exposure. And after all this training on oxycodone self-administration, if we switch them to, and it's under FR, if we switch them to fentanyl and do this dose response curve, at low concentrations, low per infusion concentrations of fentanyl, we saw increased responding in the animals who've been exposed to THC and presumably had, received this sort of downregulation, if you will, of the CB1 receptors. And so this is sort of, consistent with the story I've been trying to tell you with exogenous external THC signaling decreasing, responding because it increases the reinforcing value. Here we have potentially is particularly when there's not very much fentanyl available, they have to respond harder to feel the same signal, the same reinforcing value because of the loss of the endogenous, presumably the loss of the endogenous CB1 signaling. I just wanted to point out, this is not related to the reward value. This is the first group of animals I showed you. And this is just to point out the specificity of a given endpoint and the lasting nature of the effect. And so these are not the oxycodone trained animals. This is the first cohort. We did something super simple out into adulthood, about 168, 154 days into adulthood in those animals, which I showed you had this tolerance to the thermoregulatory effects. The females were maybe a little more sensitive, the males a little less, but it's present in both. We did a feeding assay, which is really stupid and simple. We just measured how much food they ate in a 6-hour period during the day for 2 weeks straight. And there wasn't any effect in the female animals, but at least in the male animals, the ones who had been exposed to adolescent THC, again, during adolescence, they hadn't seen this repeated THC since day 46, had a lasting behavioral phenotype. They ate more food. And that's just to sort of convince you that these effects are persisting, and that, you can we're sort of inferring that these are more or less permanent effects. And if you probe it the right way, you're basically going to see the animal being disrupted forever. Okay, I will end there. And again, thanks to CMCR for funding this work. And most of this, or a lot of this, was set up by Jacques Wynn and done by Annabelle Grant.